First question, which of the following statement is wrong about showing the Kranz anatomy? We will read one complete topic and then I will ask question from one person. Kranz anatomy. This topic. C4 pathway. Plant have adapted uh, plant that are adapted to dry tropical region. Dry tropical region means where we have less light in uh, more light intensity, dry area, correct? Water shortage. Dry tropical region. What happened to this dry tropical region? Have C4 pathway mentioned earlier. Though these plants have C4 oxaloacetic acid as the first CO2 fixation product, they use C3 or Calvin cycle for the main photosynthetic pathway. So in this case, C4 plant present in the dry tropical region. First question. Second, in C4 plant, first product is oxaloacetic acid. Third statement, in C4 plant, C3 pathway is also present, and C3 pathway is responsible for food formation. Correct? Then what is special in the C4 plant? Their leaf structure is different. Special. They can tolerate very high temperature. They can manage high light intensity, and they do not have photorespiration. That's why the productivity is also more. Two reasons: more chloroplasts, more temperature tolerance, no photorespiration. Photorespiration means waste. Manage high light intensity. So there are special large cell present around the vascular bundle. Vascular bundle means xylem phloem in C4 plant, and this large cell around vascular bundle is bundle sheath cell. You must have read this same thing in your anatomy anatomy of flowering plant. Bundle sheath cell. Bundle sheath cell present around the xylem and phloem. Okay. Leaves which have such anatomy, special anatomy, is known as Kranz anatomy. Kranz means wreath, mala, garland, right? Is a reflection of arrangement of cell. Ki how cell are arranged. Ki like you have mesophyll cell, you have bundle sheath cell, like that. Okay. Bundle sheath cell form multiple layer, several layer around the vascular bundle. Multiple layer. Bundle sheath cell form multiple layer. Very, very, very important. Bundle sheath cell form multiple layer. And NCERT also marked this important. Dark color. Bundle sheath cell form multiple layer. More number of chloroplasts. Thick wall. Thick wall is not allowing gaseous exchange, unnecessary gaseous exchange, and no intercellular space. Correct. Example of C4 plant. Maize and sorghum are two examples of C4 plant given in NCERT. Both have Kranz anatomy and there is a special distribution of the mesophyll cell. Correct. What is special in the C4 plant when it comes to the cell? Bundle sheath cell are special because in C3 plant we don't see bundle sheath. Only C4 plant have this bundle sheath. C3 में नहीं होती, C4 में होती. This pathway is known as Hatch and Slack pathway. C3 pathway is known as Calvin cycle. C4 pathway, second stack pathway. In C3 plant, primary CO2 acceptor is RUBP. Here, 3 carbon phosphoenol pyruvate. In C3, RUBP, C4 phosphoenol pyruvate. This is present in mesophyll cell. In C3 Calvin cycle, everything happen in mesophyll cell. Enzyme. Enzyme response in C4 plant is peptidase or phosphoenol pyruvate carboxylase. In C3, Rubisco. In C3, Rubisco. In C3, Rubisco. Okay. So when you read C4 plant, compare with C3. Okay. Compare with C3. Next. Where is this? Huh. Enzyme responsible for this fixation, PP carboxylase peptidase in C3 plant, Rubisco. It is important to register mesophyll cell do not have Rubisco enzyme in C4 plant. In C3 plant, mesophyll have Rubisco. C4 acid oxaloacetic acid is formed in mesophyll cell. In C3 plant, this PGA is formed, 3 carbon PGA is formed. So in C3 plant क्या होता है कि when you take RUBP, RUBP react with one CO2 and produce two molecule of PGA, phosphoglycerate. Correct. Enzyme used in this case process Lupus. Correct. So what is primary acceptor in C3 plant? RUBP. In C4 plant, PEP, phosphoenol pyruvate. What is the product in C3 plant? PGA. In C4, oxaloacetic acid. Correct. Then two other product form. What are two other product? Malic acid and aspartic acid. Both in mesophyll cell. Very, very, very important. Like bundle sheath cell form multiple layer is important. Here, bundle sheath cell form multiple layer around vascular bundle. Same here. This word is really important. Malic acid and aspartic acid both produce in mesophyll cell. Then transported to bundle sheath cell because transportation here. Next. In bundle sheath cell, C4 acid breakdown release CO2 and 3 carbon molecule. These 3 carbon molecule transported back to the mesophyll. Excuse me. Transported back to the mesophyll were converted into PEP again and cycle complete. Correct. What is the use of release of CO2? Key, if you see the cycle, CO2 will enter into the mesophyll cell, correct? With the help of phosphoenol pyruvate, it will fix and oxaloacetic acid will form, correct? Oxaloacetic acid, 4 carbon acid is transported, like malic acid, aspartic acid is transported through plasmodesmata. In bundle sheath cell, this C4 acid will break down, processes decarboxylation, removal of CO2. This CO2 used in Kelvin cycle. कि C4 एसिड बंडल स्थित सेल में गया, उसका यूज क्या हुआ? एक तो उसने 3 कार्बन का मॉलिक्यूल बना दिया, दैट विल ट्रांसपोर्टेड बैक, दैट विल अगेन फॉर्म फॉस्फेनोल पायरुवेट. और सेकंड ये था कि रिमूवल ऑफ CO2. व्हेन CO2 इज रिमूव्ड तो होगा क्या? इट इज यूज्ड इन द केल्विन साइकिल. एंड हियर Rubisco enzyme is used, and here Rubisco enzyme is present in bundle sheath cell. Rubisco enzyme is present in bundle seed cell. This is Hatch and slack pathway, C4 pathway. Hatch and slack pathway, C4 pathway. Next, CO2 release in bundle seed enter into C3 or Kelvin pathway, a pathway common to all plants. Remember, C3 cycle or Kelvin cycle is present in all the plants. All the plants. Very important point. Bundle seed cells are rich in rubisco, but bundle seed cells do not have package. Why? Because package is present in mesophyll cell. Correct? Mesophyll cell. Correct? Package have mesophyll cell. Bundle seed cell have rubisco but not package. Basic pathway result in the formation of sugar. Food. Sugar means food is Kelvin pathway. Common to both C3 and C. तो अगर खाना बनाएगा तो Kelvin pathway बनाएगा. If food is formed, it is formed by the Kelvin pathway only. In C4 plant, you know Kelvin cycle occur in mesophyll in bundle seed cell, not mesophyll cell. Jordan having network issue. Okay. In case of C3 plant, Kelvin cycle occur in mesophyll cell. In C4 plant, bundle seed cell. Anybody have question? Ask me. Anybody have question? Read this properly once again. I will ask question from this again.
Good morning, sir. Sorry, got late.
Good morning, Anupa. And also, guys, read your NCERT. Okay, first go through this, read your NCERT again and again, and then I will ask question. Let's read this photo respiration again. Photo respiration happen only in C3 plant, not in C4 plant. Photo respiration. This reaction where RUBP normally react with CO2 and produce 2 PG. This is the step occur in C3 plant normally. Correct? Rubisco is the most abundant enzyme in the world. Rubisco can interact with the CO2 or oxygen. Correct? Rubisco bind more with CO2, not oxygen. Correct? When CO2 and oxygen is exactly same amount, Rubisco bind with CO2. And this binding is competitive. What I am trying to say, कि Rubisco can bind with oxygen, Rubisco can bind with CO2. If both amounts same, तो Rubisco ज्यादा CO2 के साथ bind होता है. Why? क्योंकि CO2 के साथ affinity is more, है ना? Rubisco के साथ affinity is more. ठीक है? तो Rubisco किसके साथ bind करेगा? Like whether Rubisco is going to attach with the oxygen and CO2, all will decided by the concentration percentage. Correct? In case of C3 plant, small amount of oxygen can bind with the Rubisco. Okay. C3 plant में oxygen bind to Rubisco, तो CO2 fixation will decrease. Why? क्योंकि normally CO2 bind with the Rubisco and there is photosynthesis process. But if oxygen bind with Rubisco, then obviously photosynthesis will decrease. Photosynthesis will decrease. And RUBP in this case, when bind with the oxygen, convert into phospho PGA, normal PGA plus phosphoglycolate. Very, very, very important. Phosphoglycolate. So one phosphoglycerate normal, but phosphoglycolate is special. So the process of formation of phosphoglycolate when bind with the oxygen is photorespiration. No food formation in this case, no ATP formation. Instead, it results in the release of CO2 and utilization of ATP. ATP is wasted, no benefit. Correct? No ATP synthesis, no NADPH synthesis, only utilization. Correct? So we don't know what is the benefit of this process. Correct? In case of C4 plant, same process does not occur. Why? Because C4 plant have mechanism that increase the CO2 concentration at the enzyme site. इसका मतलब ये कि in C4 plant more amount of CO2 is available with Rubisco. Why? Because inside the bundle sheath cell more CO2 is produced. More CO2 means Rubisco will bind only to the CO2, not oxygen. Right? This takes place when C4 acid from the mesophyll cell broke down into the bundle sheath cell and released CO2 inside. What is the special in C4 plant? कि C4 plant के अंदर CO2 is released inside, so more CO2. But in C3 plant, mesophyll cell, more oxygen is present outside, so some amount of oxygen can also bind. Correct? In C4 plant, more CO2 released inside, so increasing inside cell more CO2 concentration. So this makes sure only CO2 bind with Rubisco. Okay, no oxygenase activity. Correct. So we can say C4 plant do not have photorespiration, and that's why you will say कि productivity is better in C4 plant, yield is better in C4 plant, and along with that, C4 plant can show high temperature, and you can see this. This table is extremely important. Correct. Cell type one by one. How many people are there? Anvita, Barinder, Manan, Devansh, Jordan, Nikki, Nizla, Ryan, Rishikesh, Sara, and Shokin. Anvita, cell type present in the Calvin cycle, in which Calvin cycle takes place in C3 plant. Mesophyll cell and C4 plant, bundle sheath.
Bundle sheet.
Next, Barinder, cell type in which initial carboxylation reaction takes place. Initial carboxylation means addition of CO2. Mesophyll cell in both plants.
Sir, can I come back in 2 minutes?
Okay. Then Aditya, how many
Can
cells?
you explain this again?
Yes,
The
sure.
second point, cell type in which the initial carboxylation reactions occur.
निर्जला C3 साइकिल जो कैल्विन साइकिल है ना, वो तो सिर्फ C3 प्लांट में होती है और वो मेसोफिल सेल ही होती है उनके अंदर, है ना. C3 प्लांट ओनली मेसोफिल सेल, तो कैल्विन साइकिल अकर इन मेसोफिल सेल ओनली, है ना.
Yes, sir.
बट इफ वी टेक C4 प्लांट, तो C4 प्लांट में क्या होगा, लाइक दिस. तो C4 प्लांट के अंदर तो जो फर्स्ट CO2 ऐड हुआ है वो फॉस्फोइनोल पायरुवेट के बाइंड होने की वजह से हुआ है ना. CO2 ऐड विद द फॉस्फोइनोल पायरुवेट. तो ये भी कहाँ पे हुआ, मेसोफिल सेल में ही ना. बंडल शीथ सेल में तो नहीं हुआ ना.
नहीं. Okay, sir.
हाँ, Next, how many cell type Nirjala? Next question. How many cell types does leaf have that fix CO2? कितनी cell type है C3 and C4 plant में? C3 have only
In C3,
mesophyll
one, one mesophyll.
and C4 two mesophyll as well as bundle.
Bundle sheath.
Jordan, primary CO2 acceptor in C3.
Pep. Sorry.
RUBP,
RUBP, huh?
5 carbon, right?
Yes, sir.
And in C4 plant,
It's Pep, yes.
3 carbon. Sarah, what is the product in C3 plant? First CO2 fixation product. 3 carbon PGA in C3 plant and 4 carbon
then
oxaloacetic acid
oxaloacetic acid in C4.
in C4. Next, Devans. Does plant have Rubisco? C3 plant have Rubisco and C4 plant have Rubisco? Devans. Yes. Does peptidase is present in both the plant? Rayan? Only in C4.
Only
C3
C4.
plant do not have this. Then Rishikesh.
Yes, sir.
Which cell in the plant have Rubisco? In C3 plant,
Mesophyll cells,
mesophyll. C4 plant,
bundle cells.
correct. CO2 fixation rate in a high light intensity.
Sir, medium.
In C3 plant, photosynthesis amount, like food synthesis amount, is less. In C4 plant, more.
C4.
Whether photorespiration is present as low light intensity in C3, more photorespiration in C4 plant. Doesn't matter what is the situation. In C4 plant, no photorespiration. C3 plant have photorespiration, C4 plant do not have respiration. Whether low light, high light, low CO2, high CO2 concentration. Understood? No photorespiration in C4, as simple as that. Correct? C4 plant can tolerate high temperature 30 to 40 degrees Celsius. C3 plant can tolerate less temperature 20 to 25. That's why C4 plant can present in the tough situation.
Sir,
Yes.
sir, whether photo respiration would be present at high CO2 concentration in C3 plants sometimes, right? Not
Hmm. Yes. Sometimes. No, but photorespiration is always there in C3. Why? Oxygen is also present in air, that of oxygen, 20%. So concentration of CO2 is very high
yes.
in air. Yeah.
Sir,
Example of yes.
सर क्या कहते हैं जो रुबिस्को है वो C4 प्लांट में आफ्टर डिकार्बोक्सिलेशन के बाद ही तो होगा
हाँ, तो इसलिए रुबिस्को C4 प्लांट में बंडल सीड सेल में होता है.
ठीक है.
So maize, sugarcane, sorghum, these are the example of C4 plant. All of you understood?
Yes, sir.
So, first question, Jordan, 180. Which of the following statement is wrong for showing crane anatomy? Tell me first statement one by one. Bundle seed cell.
cells are present. True. That is.
Next,
Uh,
Nirjala. Cells surrounding the vascular bundle have large number of chloroplasts.
Wrong, sir.
In C4 plant, there is more number of chloroplasts.
Okay.
and the wreath-like structure also.
Yes. Nikki Saharan. Primary CO2 acceptor in C4 plant is C4 acid. Why?
Wrong.
Wrong. Correct. Why? Because primary CO2 acceptor in C4 plant is PEP.
PEP.
C-carbon.
And that's in the
And
mesophyll cell, no, sir.
yes, that in mesophyll cell. And Ryan, Rubisco is present in stroma of chloroplast of bundle sheath cell in C4 plant. Yes,
Sir,
Rubisco is present in the bundle sheath cell. Now tell me, after reading NCERT, question look bit easier and every statement is clear cut.
yes.
नेक्स्ट प्लांट ग्रोथ एंड डेवलपमेंट कल पूछा था ना बरिंदर तुमने आज प्लांट ग्रोथ की कर रहे हैं हम ग्रोथ growth rate is the increase growth per unit time. Growth rate is increased growth per unit time. Growth rate. And we can express this mathematically. If an organism or part of organism like cell can produce more cell in different way. Example, in arithmetic growth, there is a limited growth. One cell become mature, another cell keep on dividing. One cell become mature, another cell keep on dividing. In geometric, like zygote, unlimited growth. One cell produce 2, 2 to 4, 4 to 8, 8 to 16, doubling every time. Example, if you see zygote divide initially 1 to 2, 2 to 4, 4 to 8. Initial growth is geometric phase. All cell divide. बाद में later on arithmetic phase will also come. Correct? Said that lose the capacity of division and dark color is divided. Remember, in case of embryo development, we have both growth, geometric plus arithmetic both. Correct? Next. Growth rate show increase that may be arithmetic or geometric. Like in arithmetic growth, mitotic cell division will happen. Only one daughter cell will continuously divide, only one cell, not both. And another cell will become mature. One cell divide, another mature. Right? Simplest explanation is root elongation. Root elongation at fixed rate. Correct? You know, length of the root increase very slowly. So that's why one cell mature, one cell divide. Right? We get the linear curve. We have the height of the plant and time. Length of the organ and time. Remember, in y-axis you have length of the organ and time. Time and length of the organ. Correct? Length against time. So LT after T time, what will be the length? Initial length and RT. Example, it is same way कि मान लो हमारे पास T टाइम पे हमने देखा कि लेंथ एक स्ट्रक्चर की थी 5 सेंटीमीटर. देयर वाज 5 सेंटीमीटर लेंथ at 0 time. After 10 days, what we see length is 10 centimeter. So LT is the final. This is LT. This is L naught initial. Right? What is the time? 10 days. So if we see 10, 5, R and 10, what we can see? Half centimeter length will increase every single day. Correct? Half centimeter length will increase every single day. This is known as growth rate or elongation per unit time. growth rate or elongation per unit time. This is elongation 1.5, like half centimeter elongation per day. Time can vary per day, per minute, per second, per hour. Correct. In case of geometrical, in geometrical growth, in most of the system, initial growth is very slow. Initial growth is very slow, known as lag phase. Correct. Then rapidly exponential rate log phase. Exponential log. Correct. Both progeny cell will show mitotic cell division, retain the ability of division, continuously dividing. Cell continuously dividing in exponential phase. But we have limited food. We have limited food supply, so growth will slow down and stationary phase. Final. If we plot parameter of growth, we will get sigmoid or S-shaped curve. Correct? Sigmoid or S-shaped curve. This is present almost in every living organism in natural condition. Every living organism shows this type of growth. Same in human being. Starting like 2-3 months or like starting 6 months is very slow growth for the child baby, right? After that, next 4-5 year rapid growth. Then the growth rate is very slow. Correct. Here you can take example of the weight. So equation are different. In case of length, equation was LT is equal to L naught plus RT. Correct. This was for the arithmetic growth. This is for geometric growth for weight. WT is equal to W naught ERT. Correct. Final size. And final size can be given in the different form. It can be weight, it can be height, it can be number. Correct. Same. Initial size, it can be weight, height, number. R same growth rate. Correct. T same time and E is same natural logarithm. R in this case is a relative growth rate. Means what is the speed per unit time? It may be per day. How much rate increase in 1 day or 1 second or 1 minute? It measures the ability of plant to produce new plant material. And also we can call it as efficiency. It means if R value is high, plant is producing material very fastly. If R value low, it means growth is less, efficiency is less. Correct? And we can say final value always depend upon the initial value. Same way, final money, how much you have in your pocket, it depend upon the initial one. If I give one person 1 crore rupees, another person 10 rupees, they cannot have the same final amount. It will vary. Same thing. So, how is it going, guys? Anvita,
Good, sir.
Barinder,
Yes,
Barinder,
sir.
how is it going?
So good.
Manan, is not there. Devans, Devans समझ में आ रहा, अच्छा जा रहा है?
Yes, yes, sir.
Jordan, how is it going?
Going good, sir.
Nikki,
Yes, sir.
सही समझ में आ रहा अच्छे से?
Yes, sir.
Okay. Nizila,
Yes, sir.
how is it going?
Good.
Dayan,
Yes.
Rishikesh,
Going good, sir.
Sarah
It's going good, sir.
and Aditya.
नहीं सर सही है
Huh. So, if you understood all, these are the two type of growth in plants. First, geometric growth. First, very slow lag phase. Second, exponential log phase. And last, stationary final phase. This is the ideal curve, which is also known as the sigmoid growth, present in the cell in culture or most of the structure in higher plant or plant organ. For most of the thing, this is the best graph. Correct? This for length or height, length of the root or height. Height increase slowly, length increase slowly. Correct? And these are the two curves. Arithmetic, low division. Geometric, double every time. So look at this question. Devans, what is the answer of this question? Identify the growth type in the diagram. A and B. Any doubt, guys? Anybody? Straightforward question. A is arithmetic, B is geometric. Third question. Respiratory quotient depend upon the type of substrate. Carbohydrate this, pure fat this. First cell on the earth lived in the aerobic condition and inner mitochondrial membrane has no enzyme for Krebs cycle but has for the ETS. Correct? How many statements are incorrect? पहले तो मुझे ये बताओ, how many of you face issue कि you read all the statement, then you check, oh, this statement is correct, this is wrong, this is wrong, this is wrong. So two correct, answer will be two. कितने लोगों के साथ ऐसा होता है? How many of you face this issue?
Me, sir.
Raise your hand, guys. How many of you do mistake like this? Nirzila, Jordan, Aditya, Nikki, Sara. Okay. Anvita, Rayan. तो पॉइंट ये है, this will happen, but when you do mistake, become more conscious कि अब हो गई, next time से नहीं होगी at any cost. I will solve one question less. That is absolutely fine. But if I know the topic, I should not make mistake at any cost. As simple. Correct? Solve the respiration chapter. Respiration. Respiration in plant notes. Respiration in plant NCERT. First, we'll do from the end. This part will be questioned. Ritesh Gupta. Come to the topic respiratory quotient. Respiratory quotient will tell what is the efficiency of the cycle. correct? In aerobic respiration, how much oxygen consumed and how much CO2 release, correct? Their ratio of the volume. Even trust me, volume word is important. How much CO2 is evolved, how much oxygen is consumed. It will tell us respiratory quotient or this word respiratory ratio. Sometimes people will miss this word respiratory ratio. Remember, my job in this session is not reading biology. My job is to highlight where you can make mistake. Okay. First statement, respiratory quotient depend upon, and we already highlighted this, the type of respiratory substrate used during the respiration process. So, Priyanka, first statement, respiratory quotient depend upon the type of respiratory substrate used during the respiration. Is this correct? Yes. Second, when carbohydrate used as substrate, carbohydrate are completely oxidized and RQ value will be 1. Respiratory quotient or respiratory ratio will be 1. It means equal amount of oxygen O2 are evolved and consumed. Next, Nikki. When carbohydrates are used as substrate, they are completely oxidized and RQ value is 1. Is this correct?
Yes, sir.
Yes. Third, why? Because when carbohydrate like glucose react with 6O2, 6 CO2 produce 6H2O and energy, so respiratory quotient will be 1. Correct? For fat using respiration, less than 1. Like fatty acid tripalmitin formula C51, C51, H98O6. Very, very, very important formula. C51, H98O6. They can ask you direct formula of tripalmitin. How many oxygen consumed? 145. Number of CO2 produced? 102. How many H2O is produced is easy to correlate. How? Number of hydrogen given here same. Same. Correct? Even CO2 is easy. If you know carbon is 51, double of this 102. Correct? So if you know zero point 0.7, you can even calculate this 140. But if you अगर हमें पहले ही आइडिया है तो चीजें आसान हो जाएंगी. Last, protein. In case of protein, about 0.9. Last line. What is important to recognize that living organism respiratory substrate is more than one. Correct? Example, we can have glucose, carbohydrate, we can have fat, we can have protein. So all three possible, more than one. Pure protein and fat are never used as respiratory substrate. Pure protein and fat are never used as respiratory substrate. Brinder, pure fat can be used as a respiratory substrate, but not pure protein.
So wrong.
Wrong. Correct. Wrong. Next statement. First cell on the Earth planet lived in aerobic respiration. वैसे तो ये wrong statement ही है, बस मैं check कर रहा हूँ. Hmm. Look at this. There are sufficient reason to believe that the first Cell planet live on the atmosphere lacked oxygen. Right? First cell live in the earth atmosphere lacked oxygen, but our job is to do more. So I will finish this topic. Do plant breathe? Answer is not direct. Answer is yes. Plant require oxygen for the respiration and they do CO2. तो but plant तो खाना produce करते हैं. Plant produce oxygen. Answer is that is food making process is different. Respiration is different. So plant also need oxygen for the respiration. But how plant are different from animal? Plant do not have special organ for gas exchange. Example, we have lungs. Insect have tracheal system. Animal present in water have gills. We don't have, like plant don't have any of these stuff. Correct? Plant have stomata and lentils. Right? Why? Because why special organ are not present in the plant? First, they can manage their own need. Individual cell can manage their own needs. So very less transportation of the gases. Like in our case, lung will transport to the full body, but there they have direct requirement, direct exchange. Correct. Second, their demand is very less. We are homeotherms. We need to maintain constant temperature. So if we need constant temperature homeotherm, so what will happen? In this case, we will see ki there will be less demand in case of plant. Why? Plant don't need to maintain constant temperature. प्लांट के अंदर कांस्टेंट टेम्परेचर नहीं होता, उनको जरूरत नहीं है, सो उनकी रिक्वायरमेंट भी बहुत कम है. We homeothums need lot of energy. We eat all the day. Why? Because homeostasis, energy requirement. Correct? So less demand, root stem leaves respire at lower rate than animal. Why? Because they are not homeotherms. Only during photosynthesis, large volume of gas is exchanged. Why? Active process. And each part of the leaf is well adapted to take care of on need of this, not transportation. Correct? And when they do photosynthesis, there is a lot of oxygen available, so no issue. Correct? Third reason, distance is very small. Correct? Distance is very small, so they can easily cover it. Like in human being, lungs are present in the chest region, and we have to transport oxygen to the toe tip. So in that case, distance is long. But in case of plant, distance where gases exchange is very small. Correct? Distance is very, very small. So in each living cell of the plant located quite close to the surface of the plant. Every cell is close to the surface of the plant. Correct? In case of stem, cells are organized in the thin layers inside the bark, and there is opening known as lentils. You must have seen crack lentils in plant. Lenticel in plant. These crack. So what is the purpose of crack? Gases exchange. Oh, CO2 oxygen can enter through this pore. Correct? This lentils to increase the surface area. Correct? So cell in the interior are dead and only mechanical support. You will see कि sir stem तो बहुत thick होता है. उनकी cell को कैसे, like how they will get the oxygen? Inner part of the stem is dead only for support, not for the gases. Correct? So that's why we are saying most of the part of the plant have some part exposed to air, so direct oxygen requirement. Correct? And that is why there is loose packing of parenchyma cell. in the leaf stem root. Why? Because airspace, availability of the oxygen. Correct? When glucose break down in human body or any organism, this happen. Correct? And but you know which statement is more important? Yield energy, most of which is given in the form of heat. Energy is released in the form of heat. Energy is yield in the form of heat. Even in our body, it is heat. Correct? And if we are using this energy, what is the purpose? We make another molecule that cell need. Correct? That's why plant will break down the glucose molecule. So not all the energy is released in the form of heat. Correct? Not all the energy released in the form of heat. So glucose will break down in multiple small steps. And this multiple small step will produce ATP. Right? Why? Because in case of respiration process, we have electron transport system. In electron transport system, or in total, we make 38 ATP. 38 ATP. Correct? Electron transport system 34. Right? Reason is this. Because all ATP produced in small step, not in one step. Right? During the process of respiration, oxygen used, CO2 water energy released as product. Correct? And this process require oxygen. Some cell live where oxygen may or may not be available. And you can see there are sufficient reason to believe that if first cell was lacking oxygen, means anaerobic. We know many organisms that are adapted to anaerobic condition. Anvita, tell me one organism which is adapted to the anaerobic condition given in NCERT.
Mycoplasma.
Very good. Mycoplasma. Mycoplasma is aerobic, but it can survive without oxygen. Can survive without oxygen. Anaerobic. And it can be. Some become facultative anaerobic. In special condition, they become facultative anaerobic. This is the best example. Facultative anaerobic, best example is mycoplasma because normally they are aerobic. But if there is difficult situation, become anaerobic.
So
Right?
then where do purple sulfur bacteria fall under? So they're anaerobes.
Purple sulfur bacteria. No, you are talking about in sulfur
Okay,
bacteria, they are chemosynthetic organism. They make their food in
okay,
absence
okay.
of
I got
light. That is
confused
their
again.
food.
I mean, okay, they require sulfur, yes, sir.
That is the story of the photosynthesis.
Yes,
Correct?
sir. Yes, sir.
Obligate uh, anaerobe are Clostridium bacteria, tetanus, Clostridium tetani. If there is a cut, bacteria will stuff there in absence of oxygen. That is the best example of obligate anaerobe. Right? So that's why in all the living organism there is machinery that can partially break down glucose without help of oxygen, and it is present in all living organism, and this process is known as glyco. Correct. So through this you got to know why plant do not have lot of requirement of oxygen, why no respiratory organ, what is the story, and you got question from this. So this Rayan, first cell on the earth planet lived in aerobic condition. Definitely wrong. Last, inner mitochondrial membrane has no enzyme for Krebs cycle, but has for ETS. One exception, succinic dehydrogenase. Succinic dehydrogenase or succinate dehydrogenase, same name. This is exception. Why? I will show you in diagram. Look at this diagram. Though it look messy, but what is happening in this diagram? There is one common thing. What should be the color? Every color I used here. See, succinic acid convert into fumaric acid. Correct? Enzyme used in this process is succinic dehydrogenase. Same thing here. Second complex is succinate dehydrogenase complex 2, where succinate convert into fumarate. Correct? And this enzyme is present in inner mitochondrial membrane. Succinate dehydrogenase is present in inner mitochondrial membrane. So this is the enzyme of Krebs cycle. सक्सिनेट डिहाइड्रोजन किसका एंजाइम है? Krebs cycle का, लेकिन कहाँ प्रेजेंट है? Inner mitochondrial membrane. Can we say this statement is wrong? Inner mitochondrial membrane has no enzyme for Krebs cycle. Wrong. Succinate dehydrogenase is the enzyme of Krebs cycle, but present in intermitochondrial. But definitely, inner mitochondrial membrane have all the enzyme for electron transport system. Complex 1, complex 3, 4, 5, everything present in electron transport system in the membrane. Any doubt, guys?
Yes.
So, when you see this question, see first two options are correct, last three wrong. So you will try to mark, okay, answer is two statements correct. But they asked,
So could I be back in 10 minutes? So I have to go breakfast.
okay, Jordan. Okay, but actually, answer will be 3.
3, sir.
You have to be very conscious what is the demand of the question. You cannot make mistake. Like here, it's okay. That's why you are writing full syllabus test. When yesterday you wrote full syllabus test, you understood, okay, I will make mistake like that, and I should not repeat those mistakes again at any cost. And in that process, you imagine, okay, whatever happen, I will not make mistake in correct, incorrect at any cost. Because if you don't know topic, that's okay. But if you know topic and still make mistake, that is horrible, pathetic, and you feel guilty. Am I right, guys? क्या मैं सही बोल रहा हूँ?
Yes.
Raise your hand. Yes, sir. He feel guilty and ashamed, to be honest, कि when making mistake, और हमें पता है कि वो क्वेश्चन आता है, वो टॉपिक अच्छे से आता है. ठीक है, तो नेक्स्ट टाइम मेक श्योर कि ठीक है, आई विल डू 1 or 2 क्वेश्चन लेस. जितने आते हैं वो तो अच्छे से हो. वट एवर आई नो, इट शुड बी परफेक्ट. एंड दैट्स वाई वी आर रीडिंग एनसीईआरटी. कि इन बायोलॉजी वी कांट मेक मिस्टेक. In biology, we'll also see how we are making mistakes. Okay. Question. Next question. One 78 done. 177. Effect of water as a factor is more through its effect on the plant rather than directly on the photosynthesis. Let me take this topic. Photosynthesis in higher plant. In last class, we left last topic. Factor affecting the photosynthesis. Factor speed of the photosynthesis is very important because we want to produce more product even in the crop plant. Correct. There are multiple factors. Internal factor is known as plant factor or external factor. Internal factors are like what is the number of leaf, size of leaf, age of the leaf, orientation of leaf, mesophyll cell, chloroplast, internal CO2 concentration or amount of chlorophyll. These plant will decide this. Plant leaf will decide this. Chlorophyll amount will decide this. Mesophyll cell will decide this. Correct. These internal factor or plant factor are dependent on the genetic predisposition and growth of the plant. Genetic means जैसे हमारी ग्रोथ कितनी होगी डिपेंड अपॉन आवर जेनेटिक मेकअप सेम प्लांट हाउ प्लांट विल ग्रो इट विल डिसाइडेड बाय जेनेटिक मेकअप करेक्ट एंड हाउ प्लांट ग्रो बेस्ड ऑन दैट यू कैन टेल हाउ मच photosynthesis occurs. External factor sunlight temperature external CO2 water correct and all these factors will act simultaneously very important word simultaneously at same time.
Sir, we'll be back in 2 minutes.
Okay, sure. So most of time one factor is the major factor that affect the rate speed. Hence, at any point it determine which level is suboptimal. Suboptimal मतलब जितना चाहिए उससे कम. Whatever is required less than that is the suboptimum level. So there are multiple factors that affect the process, and Blackman gave this law in 1905. Blackman gave law of limiting factor. Correct. And what he said, if you have a chemical process affected by more than one factor, then rate will be decided by the factor that is nearest to the minimum value. And it is the factor that directly affect the process if its quantity is changed. एक example देता हूँ. You want to make a house, a big home. Correct. You have money, you have labor, but you don't have land or very short land, like very small land, not short, very small land, like 100 square feet is available, but you want to make bungalow. Not possible. Why? Land shortage you have. So land is the suboptimal factor. Land decide करेगी कि घर बनेगा कि नहीं. Now land is the limiting factor. Second, you have good amount of land. What is required for making bungalow? But labor is not available. Like half labor is available. So what is the limiting factor now? Labor. Labor is the main factor that will decide whether work will progress in the perfect time और ज्यादा टाइम लगेगा. लेबर डिसाइड करे है ना. और यू मस्ट हैव सीन कि मोस्टली जो हम प्लान करते हैं उस टाइम में कंस्ट्रक्शन रेडी नहीं होता. करेक्ट. थर्ड ऑप्शन की लेबर इज अवेलेबल लैंड इज अवेलेबल बट वी हैव शॉर्टेज ऑफ मनी. ये भी होता है कंस्ट्रक्शन के टाइम में. तो दैट इज वन प्रोसेस. अंडरस्टूड. तो this is how like limiting factor means shortage. Whichever value is less will decide. सब कुछ परफेक्ट है एक चीज की कमी है तो वही डिसाइड करेगी स्पीड क्या होगी. सेम एग्जांपल गिवन हियर लाइक we have green leaf, we have proper light, we have CO2, but temperature is low. What will happen? Photosynthesis will be low. If temperature will become normal, photosynthesis become normal. Correct. First factor light. Correct. So there is a linear relationship between the incident light and CO2 fixation rate at low light. If less light, less CO2, sorry, less light, less photosynthesis. More light, more photosynthesis. तो लीनियर रिलेशनशिप होती है ना. बट इन लो लाइट कंडीशन लाइट बहुत कम है, लाइट बढ़ाते जाओगे फोटोसिंथेसिस इंक्रीज हो जाएगा. एज सिंपल है. हाई लाइट इंटेंसिटी में रेट बिकम ऑलमोस्ट फिक्स. क्यों? लाइट तो ऑलरेडी है ही. वी ऑलरेडी हैव लाइट, सो लाइट इज नॉट शॉर्टेज. करेक्ट. तो फर्दर इंक्रीज नहीं होता. करेक्ट. other factor become
So,
limiting.
this graph can be said as the sigmoid one.
What?
In the geometric phase, we see that it is the sigmoid S curve. So this one
हाँ.
also can be.
नहीं, बट ये जो कर्व है ना, वहां पे कर्व
Yes,
ग्रोथ की बात कर रहे थे हम, यहाँ पे ग्रोथ की बात नहीं कर रहे हैं, हम तो फोटोसिंथेसिस की बात कर रहे हैं, मोर और लेस उसी के टाइप का होगा.
yes.
नेक्स्ट, लाइट सैचुरेशन अक्कर एट 10%. इसका मतलब फुल लाइट का अगर 10% भी अवेलेबल है, इफ यू हैव इवन 10% ऑफ द फुल सनलाइट, दैट इज मोर देन सफिशिएंट टू रन द होल यूनिवर्स. नॉट होल यूनिवर्स, होल अर्थ, है ना. 10% भी सफिशिएंट है. दैट्स व्हाई मोस्ट ऑफ टाइम यू विल सी कि we don't have shortage of light. We don't have shortage of light. So light is not a limiting factor. लाइट की कमी नहीं है. कमी नहीं है तो लिमिटिंग फैक्टर हो नहीं सकता. एक्सेप्ट प्लांट लिव इन द शेड एंड डेंस फॉरेस्ट. क्यों? क्योंकि अगर डेंस फॉरेस्ट है, अमेजोन रेन फॉरेस्ट है, वहां पे जो ग्रास है, ग्रास तक तो लाइट पहुंच ही नहीं रही प्रॉपर, तो दे माइट हैव शॉर्टेज. बट नॉर्मली तो बहुत सनलाइट है, बहुत गर्मी है ना, तो दिक्कत नहीं है. ठीक. Increase in incident light beyond a point might break down the chlorophyll and decrease the photosynthesis. अगर बहुत ज्यादा सनलाइट होगी, जैसे होता है ना एकदम एक्सट्रीम हीट, तो वहां पे भी तो लीफ भी तो बर्न हो जाएंगे. लीफ बर्न मीन्स फोटोसिंथेसिस लेस, जो फॉरेस्ट फायर है वो इंक्रीज हो जाएगी, एंड ऑल दिस. सेकंड, CO2. If question asks what is the main factor where we have shortage CO2, even this is not shortage right now, correct? Because CO2 amount is very low in atmosphere, and if we increase up to 0.05 this amount CO2, photosynthesis will increase. Why? CO2 की शॉर्टेज है. CO2 को बढ़ा दोगे तो photosynthesis भी increase हो जाएगा. If you increase the CO2, photosynthesis will increase. But after this level, it will decrease. Right? Example: in low light condition, no group respond to high CO2 condition. क्यों? Light is the limiting factor now. अगर लाइट की कंडीशन लो है, it means light is the limiting factor, not CO2. So no one will respond, and this statement is very 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 important. ठीक है. यहाँ पे लाइट की शॉर्टेज है. अगर लाइट एकदम परफेक्ट है तो C3 C4 प्लांट बोथ विल इंक्रीज द फोटोसिंथेसिस स्पीड. बट C4 plant can show photosynthesis even maximum 0.06, sorry 0.36%. C4 need very less CO2, उसमें भी वो एडजस्ट कर लेते हैं. बट C3 require more than 450, ना. their saturation will be more than 450. इसका मतलब ये है C3 की डिमांड बहुत ज्यादा है CO2 की. ओनली देन इट विल शो मैक्सिमम फोटोसिंथेसिस. उतनी डिमांड पूरी नहीं हो सकती, है ना. और C4 की डिमांड बहुत लो है. 360 में भी वो बहुत अच्छा काम करके दे रहा है, मैक्सिमम सैचुरेशन आ रहा है. तो विच वन इज बेटर? ऋषिकेश, कौन सा बेटर है ऋषिकेश? Of course C4, because they need less CO2. Correct? The fact is that C3 can respond to high CO2 concentration by increasing the rate of photosynthesis and more productivity. And we can use this by using ग्रीन हाउस क्रॉप. जैसे टोमेटो और बेल पेपर का हमने फोटोसिंथेसिस इंक्रीज किया है बाय यूजिंग ग्रीन हाउस, राइट. सो उसमें क्या बेनिफिट हुआ है? वी इंक्रीज CO2 concentration in the atmosphere, so more yield, and we applied this to tomato and bell pepper. I think in last paper this was the question, right? Tomato and bell pepper. Bell pepper was the option, I guess, in last test, biology test. Temperature, dark reaction are controlled by enzyme, and enzyme is always controlled by temperature. Chemical kinetic chapter of chemistry. Speed of the reaction controlled by the temperature. Correct? Even light reaction is also very sensitive to the temperature. But dark reaction have more enzymes, so more dark reaction is more affected by the temperature. Correct? C4 plant can tolerate high temperature, so they are not affected and they show high rate of photosynthesis. But C3 plant need very less temperature. अगर उसको ज्यादा बढ़ाने की कोशिश करेंगे, if we try to increase the temperature for C3 plant, they will be damaged. Correct? So temperature optimum for photosynthesis depend upon their habitat, where they live. Correct? Tropical plant जैसे इंडिया में, so they can tolerate high temperature, but plant in cold country like Germany, Canada, they adapted to very low temperature only. Like Himalaya plant cannot live in Rajasthan. Why? In Himalaya region, temperature is low. In Rajasthan, temperature is high. So every plant adapted to different temperature. Example, Jordan, you are there.
Yes, sir, yes, sir.
So Jordan, you are adapted to Bangalore temperature. Now you go to Kerala. So your temperature optimum is different.
Yes, sir.
So you will face issue in
Yes,
Kerala,
yes, sir.
in Pala city, right?
Huh.
Exactly. That's what temperature role is. Understood well, perfectly
Got
without
it, sir.
any doubt.
What about?
Yes. Last, from where we got question. Water. See, this statement. Even though water is one of the reactant of the light reaction, effect of the water as a factor is more through its effect on the plant rather than directly. तो plant water is affecting plant in general. Plant को in general affect करेगा, photosynthesis को directly affect नहीं करेगा. And now look at this statement. So Sarah, now tell me this sentence. Effect of water. Okay? Effect of water as a factor is more through its effect on the plant rather than directly affecting the photosynthesis. Is this correct?
Yes,
Yes.
sir.
Second. Second, water stress. Water stress will close the stomata, so it reduce CO2 availability. Less CO2 availability, it will water stress will wilt the leaf like curling, reduce surface area, reduce metabolic activity, curling. So overall, everything will be less. Leaf will shrink. Correct? Leaf will shrink. CO2 availability will be less. Stomata will be closed. CO2 availability will be less. So Anvita, second statement. Very less amount of absorbed water is used for in the photosynthesis, and any water stress cause stomata close, reduce CO2 availability, and make leaf wilt. Is this correct?
Yes, sir.
Yes. So what will be your answer?
Sir, option 1.
Option 1. Why? Water is affecting the plant direct overall. How? Because it is affecting the stomata, it is affecting the leaf in general. Yeah, understood. How many of you made a mistake in this 176 question?
सर 5 मिनट चाहिए ब्रेक के लिए, मैं चले जाऊं?
बिल्कुल. Even I think let's take break for 15 minutes, guys. I'll see you at not 15 minutes, 20 minutes. I'll see you at 9. I'll stop recording here. This question. A biochemist isolate and purify various molecules needed
Have
for
you shared
DNA.
your screen?
I am not sharing screen.
No.
A biochemist isolated and purified various molecules needed for DNA replication. When he adds some DNA, replication occurs. Means DNA replication is happening normally. Each DNA consists of normal strand paired with extra segment of DNA, a few hundred nucleotide longer than the normal. What has he probably left out of the mixture? Answer of this question is DNA ligase. Why? Because I marked this correctly, so that's why I can explain you. Not because I know from the answer key, but logically, if there are extra DNA segments, it means there were many DNA fragments like this, and these DNA fragments are joined with the help of DNA ligase. So there is a extra length. Correct? As simple as that. DNA polymerase means overall it will multiply, overall amount increase. But it is saying, so you can guess, ki, sir, DNA polymerase is also one option. Yes, it is option, but each DNA consists of a normal strand. Normal strand have with numerous segment of DNA, nucleotide long. So nucleotide, extra nucleotide are joined with the help of DNA like. तो जो हमने एक्स्ट्रा न्यूक्लियोटाइड डाले हुए हैं, बाय मिस्टेक वी एडेड मोर डीएनए लाइगेज, तो वो क्या करेगा, ज्वाइन कर देगा सबको, तो एक्स्ट्रा लेंथ बन जाएगी. मेक सेंस? Nucleotide Okazaki fragment is okay, but if you are not adding DNA ligase, how it will add extra? To join Okazaki fragment, to join nucleotide, we need DNA ligase. Make sense, guys?
Yes, sir.
Anybody have question? Ask again.
So the polymerase is for the formation of the DNA, and then after that, the ligase comes into place for attachment. No.
Yes, Jordan.
So I was just asking the order of how it goes. So like, okay, DNA polymerase has to be there in order for the DNA to be formed, like the new strand, and then ligase is for it to be joined to like the parental strand.
Okazaki fragment.
So
Join
DNA
Okazaki fragment.
polymerase causes Okazaki fragments. Okazaki fragments require DNA ligase for it to be joined.
Yes.
Okay, sir. And nucleotides are just adenosine and all that, huh?
Hmm.
ACGT.
Yes. Very basic, but still we'll do this. What is your answer? All of anybody can answer.
So obviously four
Original replication.
Hmm.
Option 4.
We did DNA. Biotech recently.
Biotechnology principles and processes.
Uh, so
Can you send biotech chapter 2 questions? Because I did the chapter at night yesterday.
sure, Anita. See, now you will understand this easily. Keep plasmid and bacteriophage. These have ability to replicate with inside the bacteria, and their replication does not depend upon the bacteria main DNA, genetic DNA. Correct? Bacteriophage is a virus that can multiply with high number in bacteria cell. Correct? High copy number. Plasmid can have two copy, or sometime maybe 50 to 100 copy. So speed of multiplication of bacteriophage is way higher than plasmid. That's what it is trying to convey, or it is can go even higher, more than 100 also. If we link our DNA with either bacteriophage or plasmid, what will happen? These two will multiply in bacterial cell. Our DNA will also multiply in bacterial cell. Okay, our DNA will also integrate with bacterial cell. So, what should be the property of this bacteriophage or plasmid or any other vector? So, we should be easily able to link our DNA to the vector, and it should able to tell us whether we are attaching our DNA at the right place or not, recombinant and non-recombinant. Easy to link and to check whether we are doing right process or not. Right process is recombinant, wrong process is non-recombinant. What should be the property of vector? First, sequence where DNA replication start and any piece of DNA can be linked and it replicate it and support high copy number is origin of replication, correct? Ori. It support high copy number, control copy number, replication start here. Correct. Selectable marker. Vector should be a good selectable marker. Why? Because it should tell us whether we are allowing only transformant growth. Because transformant means a bacteria that have external DNA, rDNA, transformant. Non-transformant bacteria do not have external DNA. Bacteria do not have rDNA. So no use of such bacteria. So our vector should be able to identify that transformant, non-transformant. What is transformation? When we introduce any DNA in the bacteria. Correct? Normally, like antibiotic resistance gene, ampicillin resistance gene, tetracycline, kanamycin, and chloroamphenicol. These four antibiotic resistance gene we can add, and these can be considered as important selectable marker for E. coli. In E. coli bacteria, we can add this artificially because normally bacteria do not have all these genes against antibiotic. Normally, we are adding. Why? Because we want to allow bacteria to take up our DNA at right place so we can grow our DNA, as simple as that. Correct. Third, cloning site. When we have external DNA, vector should have one recognition site, not multiple. Because if you for one DNA, one site, good, perfect, specific, no problem, no complication. Enzyme can easily act. Correct. But if you have more than one recognition site, it will complicate the process because multiple fragments. Enzyme don't know where to act. Correct. That's why better to have one recognition site. So enzyme can act perfectly. Example. If we have two antibiotic resistance gene, tetracycline resistance gene and ampicillin. So for tetracycline, we have BAM H1 site and SAL1. Best. B, BAM H, S, SAL1 and T, tetracycline. Best. Correct. Ampicillin PST1, PVU1. If we add our DNA at tetracycline site here, what will happen? Bacteria will die in tetracycline antibiotic. Why? Now this site is not acting, that's why. It means this will be recombinant. Understood? अगर हम बैक्टीरिया को एंटीबायोटिक में डालें और हमने डीएनए में डाल दिया and bacteria die means now our DNA reached to the right place, that's why it is inactive. Insertional inactivation, when we insert our DNA, tetracycline site become inactive. Insertional inactivation. Any doubt? This topic is done. Rho is controlled by PVU2 and Rho code for protein that is required for the plasmid replication. What are the enzyme can act on ampicillin resistance? Tetracycline is extremely important. PBR322 is the vector of E. coli bacteria and PBR322 is artificial vector. Correct? Cloning vector but artificial. EcoR1, CLAV1, HIND3 act on the same site here. What question can be asked? Select odd one out. Option will be EcoR1, CLAV1, HIND3, BAM-H1. Answer will be BAM-H1. Why? Because first three are acting at the same place. Here. Last one, BAM-H1, acting in the different place. Well, makes sense. In PBR, P stands for plasmid, B and R scientist name, Bolivar and Rodriguez. But when we do this process, it is complicated. We want to do color method. Color method is easy to differentiate. Correct? Correct? Color method, very easy. So we want to produce color in the presence of specific substrate. If you have a recombinant DNA, insert in the enzyme beta galactosidase, what will happen? Gene will inactive, insertional inactivation, no synthesis of the enzyme, and in this case, color of the colony will be white or no color. White or no color. But if normally there is blue color if no DNA and white color if you introduce DNA, and this is recombinant. Any doubt, guys? What is recombinant? With DNA, and recombinant will be white. The simplest way is recombinant will be white or colorless and non-recombinant will be blue in color. So easy to identify based on the color. Any doubt? NEET 2025 question. Okay, 2025 question. Last. We learn the process how to transfer gene from the bacteria or virus. Because here eukaryotic cell convert into different form. Example, Agrobacterium tumefaciens. This is bacteria. This bacteria affect dicot plant. And this bacteria transfer one DNA known as tDNA. What is the benefit? Normal cell will become tumor. What is the benefit for the bacteria? From this tumor cell, bacteria will get chemical for growth. Same job virus do. Retrovirus in human being convert normal cell into cancerous cell. Cancerous cell, they will get the multiplication process, replication of their genetic material. Correct? Here you have tumor-inducing plasmid of Agrobacterium tumefaciens. If we can modify this, we can transfer our gene, what we want to transfer in plant, without plant is being affected. So plant को कोई problem नहीं होगी. We are transferring our gene and plant is not having tumor. So it is beneficial for us. We can use bacteria. हम bacteria को use करेंगे अपने purpose के लिए. Correct? And plant में disease भी नहीं होगी. Correct? Still we can deliver our gene. Same thing we can do in case of retrovirus. हम retrovirus को disarm कर सकते हैं. We can transfer into the animal cell. So for plant cell we can use Agrobacterium tumefaciens. For animal cell we can use retrovirus. Correct? The question was very easy, but this topic revision was crucial. This is out of NCERT. This is out of NCERT. Answer is core zone. What is core zone? Core zone is the zone in the biosphere reserve जहाँ पे कोई activity allowed नहीं होती है ना. Like there are different zone of the biosphere
There
reserve.
are three zones, right? One is core zone, buffer zone, and another was
Yes, core zone, buffer zone, transition zone.
transition zone or manipulation zone, something was there, right?
Biosphere reserve zone. Zombie. Okay. And biosphere reserve.
So the inner one is core zone, then comes buffer zone, then comes transition zone.
Yes, that's what I said. That's what Ryan asked. Key core zone where no activity allowed. Why? Because in core zone, like tiger live, main animal live. They can attack us, and their like conservation is really important. Correct. Buffer zone where you can see like there can be some settlement of human being. Photography is allowed for education purpose. You can visit like in safari. You go to the some area outskirts just for visiting in the national park. And last transition zone where most of the activity is normal, but we have to be more careful. Okay, so these are the zone of the biosphere region. Okay, no activity in core zone, nothing, because core zone is very like, dangerous for the human being, dangerous for the animal. Why? Because they are affected ultimately. Understood? That- So core zone innermost, most protected, untouched, like national park and sanctuary, legally secured, no human intervention. Buffer zone middle, some human activity is allowed for research, education purpose, training purpose. Transition outermost, most of activity can be done here. Most of, but with the help of the local, not like any random activity. So these are the three zones, correct? Buffer, transition, and core. Innermost core, then buffer, then transition, then safest. This question. In lack of iron, when repressor protein bind to the operator, then what happens? Let me show you molecular basis of inheritance. Molecular molecular basis of inheritance. Molecular basis of inheritance will directly go to lack of iron. Lack of iron we did recently, very important. And lack of iron. Lack of iron was explained by genetist Jacob and chemist Monod. Genetist Jacob, genetic Jacob. They were the first to explain transcriptionally regulated system. Transcription, how transcription explained and how it is regulated. First one. In lack of iron, lac stand for lactose. And this lac operon is a polycystonic structural gene regulated by the common promoter and regulatory gene. What is the meaning of polycystonic structure gene? A gene that have like multiple cystone and polycystone condition, sorry, polycystone condition structure gene, polycystonic structure gene present only in case of bacteria or prokaryote. In human being or eukaryote, monocystonic gene structural gene present in the eukaryote. Understood? So we discuss any operon in bacteria only. Same lac operon is a polycystonic structural gene, and this lac operon have one common promoter and one regulatory gene. This arrangement is very common in bacteria and called as operon operating system, operated by regulatory gene promoter, right? And there are many operon like lac operon for lactose, trypsin, trypoperon, uh, histine, operon, valine. Right? Most of time these are the amino acid name. Correct. In case of lac operon, there is one regulatory gene. We said there is a regulatory gene, one regulatory gene, and this regulatory gene is I gene. I gene. Correct. I do not refer inducer. Instead, I come from the word inhibit. I is the regulatory gene and inhibitor. Why? It inhibit the process when bacteria is not present. याद रखना ये लैक ओपरोन तभी काम करता है जब खाना हो बैक्टीरिया के आसपास. What is खाना? What is food? Lactose is the food for the bacteria. If खाना present है around the bacteria, तो बैक्टीरिया के अंदर एक प्रोसेस एक्टिवेट हो जाएगी जो उस खाने को डाइजेस्ट करेगा, एब्जॉर्ब करेगा. If food is present in the surrounding of bacteria will develop a mechanism that will, what should I say, that will support the process of breakdown of this food and entering of this food inside the body of bacteria. Any doubt, guys? The regulatory gene, I gene, inhibitor. Because if food is not present, then no activity should happen, no wastage of energy should happen. Correct. 3 structural gene, 1 regulatory, 3 structure. Structure ZYA. I gene will code for the repressor in the lactoperon. Z, beta galactosidase. What is the role of beta galactosidase? Breakdown of lactose into glucose and galactose. Correct. Y gene, permease, increase permeability of the cell to beta galactosidase. Increase permeability of cell to the beta galactosidase. Correct. Because beta galactosidase will ultimately enter, like activate and break down the lactose. तो अगर लैक्टोज को तोड़ना है, वी हैव टू ब्रेक लैक्टोज, तो हमें चाहिए बीटा ग्लैक्टोसाइडेज, है ना. और बीटा ग्लैक्टोसाइडेज की परमेबिलिटी इंक्रीज करने के लिए परमेज एंजाइम चाहिए. एंड लास्ट, ए जीन, कोड फॉर ट्रांसएसिटाइलेज. व्हाट इज द रोल ऑफ ट्रांसएसिटाइलेज? वी डोंट नो. ओके, ऑल 3 जीन प्रोडक्ट इन द लैकोप्रोन, तीनों की हमें जरूरत है. व्हाई? बिकॉज we need to break the lactose, we need to absorb lactose inside the bacteria body. In other operon also, gene are present. Why? Because they are related with some activity, some metabolic activity. Look at the diagram. I gene, regulatory gene, I gene. This I gene will form repressor mRNA, then repressor protein. This repressor protein will bind to the operating system, operon. Kab? When no food. No food means no lactose. No food means no lactose. तो ये क्या होगा? Repressor will bind with the operating system. खाना ही नहीं है तो क्यों ही एक्टिवेट करना है प्रोसेस को? Prevent RNA polymerase from transcribing the operon. तो आगे कन्वर्जन नहीं होगा. RNA polymerase will not act. RNA will not form. Correct. But if food is present, it means inducer means lactose, food. If food is present, we have to stop the mechanism of prevention. How to stop? Food itself will stop the process. तो रेगुलेटरी जीन अभी भी वर्क करेगा, इट विल स्टिल वर्क, इट विल स्टिल प्रोड्यूस रिप्रेसर जीन, बट जो फूड है ना वो इसको इनएक्टिवेट कर देगा. नाउ योर ZYA जीन विल एक्ट. Z will form beta galactosidase, Y permease and transacetylase A. तो यहाँ पे पहले होगा ट्रांसक्रिप्शन, लैक mRNA एंड देन प्रोटीन परमेज ट्रांसएसिटाइलेज बीटा लैक्टोसाइड. जब खाना प्रेजेंट हो लैक्टोज is the substrate for the enzyme beta galactosidase. Make sense? Yes. Beta galactosidase will act on the lactose and it regulates switch on and switch off. That's why lactose is inducer. Lactose is inducer. If you don't have proper carbon source, like we don't have glucose, if lactose is provided in the medium, lactose can be transported into the cell through the action of the permease. अब सोचो कि most of time तो bacteria need glucose. अगर ग्लूकोज नहीं है, if glucose is not present, then we can switch to the lactose, है ना. ये स्पेशल सिस्टम है ना. if food is not present, glucose is not present, तो हमें कुछ और ऑप्शन देखना पड़ेगा. तो lactose we can utilize because ultimately lactose will convert into glucose, है ना. है ना. Next. Very low level of lactoferrin should be present in the cell all the time. कुछ एक्टिविटी तो हमेशा होनी ही चाहिए, वरना लैक्टोज कभी एंटर ही नहीं कर पाएगा. अब सोचो लैक्टोज की एंट्री पहले होगी तभी तो लैक्टोज रिप्रेसर के साथ बाइंड करेगा और तभी इसकी एक्टिविटी बंद हो जाएगी ना. इफ देयर इज सम अमाउंट ऑफ permeability, what will happen? A small amount of lactose will enter into cell, stop the repressor gene. If I gene repressor gene is stopped, then remaining process will start, है ना. If imagine there is, अगर हमने पहले लैक्टोज की एंट्री ही नहीं करवाई, ये कभी एक्टिवेट ही नहीं होगा. If lactose is not entered, then no activity at all. No lactose will enter inside the body, है ना. तो यही इसने भी बोला है. There should be low level of expression all the time. Otherwise, lactose cannot enter inside the cell. Lactose will activate the process और आगे की further process हो जाएगी. जैसे repressor gene is synthesized by all the time. Repressor always from IG. Repressor protein bind to the operating region. Repressor protein bind to the operating system, as you can see here. This one. O, operating region, repressor will bind. Correct? And prevent RNA polymerase from transcribing the operon. So RNA polymerase काम नहीं करेगा and transcription will not happen. In presence of inducer, inducer can be two. It can be lactose or allolactose. Inducer can be lactose or allolactose. Repressor will be inactivated by inducer. RNA polymerase will act to the promoter region, access to the promoter and transcription हो जाएगा further. तो जो लैकोप्रोन का रेगुलेशन है वो एंजाइम सिंथेसिस है एक्चुअली बाय इट्स सबस्ट्रेट. व्हाट इज दिस? इट इज एक्चुअली a regulation of the enzyme synthesis. Which enzyme synthesis? Beta galactosidase by Z, permease by Y and transacetylase by A. तो अल्टीमेटली क्या है लैकोप्रोन का रेगुलेशन की एंजाइम बनना चाहिए इन प्रेजेंस ऑफ सबस्ट्रेट कि व्हेन लैक्टोज इज प्रेजेंट, है ना. हाँ, ग्लूकोज और ग्लैक्टोज काम नहीं कर सकते एज ए इंड्यूसर इन लैकोप्रोन. क्यों? लैकोप्रोन तो बहुत स्पेसिफिक है ना. लैकोप्रोन इज ओनली फॉर लैक्टोज, फॉर नथिंग एल्स. इफ glucose or galactose are present already, why do you need lactose mechanism? You don't need. You need lactose only when glucose galactose are not present, है ना. So they cannot act. Why? Very specific. How long lacopron would express in the presence of lactose? Lactose की presence में तो express करेगा ही. Lacopron will work as long as lactose is present. This activity by the repressor, because repressor all the time inhibit the activity. That's why this is negative feedback mechanism, negative regulation. Correct? And there is positive regulation also, but we don't have in our syllabus. Tell me, guys, किसी को कोई doubt? Anybody have question from this topic? I can explain again, but this is extremely important. Lack of protein. You have lactose in surrounding of bacteria. Lactose is a kind of food for bacteria. Bacteria want to consume this food. That's why bacteria need some machinery. Bacteria need machinery to maintain this. और उस मशीनरी को ही हम बोलते हैं लैकोफ्रॉन. That machinery is known as the lactoferrin, where full system acts so that lactose will be absorbed. Any doubt? Every single person understood this? Raise your hand. Shokeen. Anvita. Sara. Yes. Barinder. Kshitij Gupta. Yes.
Yes, sir.
Jordan. Yes. Nezla. Yes. Barinder.
Could you
Yes.
just check
Manan.
WhatsApp, sir?
Yes. Manan. Devansh. And Nikki, मेरी आवाज आ रही है? I think she can't hear me. She is not there, I guess, right? Jordan?
Who,
Oh.
sir?
A last person, Nikki Saran, is not there. That's why it is not showing anything.
No, sir. She's not there.
If you understood everything, Aditya, tell me this. Tell me this. Whom I asked?
हाँ जी सर.
Aditya. हाँ.
Option 2 sir.
Option 2. In lack of proton, when repressor protein bind to operator, RNA polymerase cannot transcribe the operon. बिल्कुल सही बात है. Repressor protein bind to the operator and then it cannot transcribe the operon, like further process cannot happen. Right? Option 2. But I think what was the answer given in the answer key? Check once again. Question number 173. Yeah, it is 2.
Two.
Yeah. बहुत easy question था ना कि lack of operon, repressor protein bind to the operator and no further transcription. Why? Because in this direct this line, repressor bind to the operator even prevent RNA polymerase from transcribing the operon. As simple as that. Right. Okay. Next question. If you see this question, very easy, but at the same time very popular. Most of female in young age know, especially in the working women nowadays. It's important कि if there is work, important meeting, and like there is a lot of pain, like it's unbearable pain for 1 or 2 days sometime, especially first uh, day or second day. So to avoid this, on the like occasionally, female take some medication. Remember, these are not the contraceptive medication. These are not the contraceptive medication. These are these basically medication to postpone the bleeding. So once like fertilization happen, but pregnancy will continue. So these are not contraceptive. So this female goes to doctor. She asks after 4 days there is a menses and she want to postpone the bleeding because there is some work. Which hormone doctor will advise to take? Of course, progesterone. Why? Because if there is more progesterone, then uh, bleeding will not happen. More progesterone, uh, this last phase will stay longer.
The endometrium lining will still
Luteal phase.
stay together, so that's
Yes,
what the corpus luteum helps in maintaining like the endometrium lining because progesterone is produced.
correct. Human reproduction. Where principle of inheritance? Where is this chapter? Sexual reproduction in flowering plant.
I'll be back in 2 minutes.
Okay, Jordan. So I will open NCERT, new NCERT. Add to notes. Human reproduction NCERT. Okay, come to this cycle. Menstrual cycle. Reproductive cycle in the female primate, especially monkey, apes, and human being, all are primates, is known as menstrual cycle. Right? First, menstruation begin at the puberty, and this first is men arc. Men means menses, arc means starting, beginning. In human female, menstruation repeated on an average these days, and it can vary from person to person. This whole cycle event start from one menstruation and until next menstruation is menstrual cycle. Now you'll feel कि sir ये तो normally है. What is special in this case? This word is special. From where it start? From where the process start? Menstruation. One ovum is released. Ovulation happen during the middle of the process. Middle means 14th day. 14th day. 14th day ovulation will happen. 14th day. Correct, middle. Majority are shown in the diagram. Cycle start with the menstruation phase. Cycle start with menstruation phase and menstrual flow. Bleeding will happen. How many days? 3 to 5 days. But in reality, it can vary from 2 days to 1 week, 8 9 days. It depends from person to person. Menstrual flow result in the breakdown of the endometrium lining. Correct? Endometrium lining. And its blood vessel which form liquid, this liquid will come out through the vagina. Right? So bleeding will happen and this blood will come out through the vagina. So menstruation occur only when ovum is not fertilized. I discussed many times कि बहुत लोगों को इतनी भी अवेयरनेस नहीं होती कि अगर किसी लड़की में पीरियड्स आते तो उनको लगने लग जाता है कि नो दिस इज नॉट नॉर्मल बिकॉज I felt it in my childhood, like when I was in class 12th. So, next, Lack of the menstruation may be pregnancy, but not always. If menstruation is not there, maybe there, like mother or female can be weak, correct? There may be hormonal imbalance, correct? Stress, poor health can be the reason. After menstruation phase, first phase is menstruation phase. Second phase is follicular phase, also known as proliferative phase. Correct? During this phase, primary follicle will convert into the mature follicle, and in the same time, endometrium lining will regenerate through proliferation process. That's why also known as proliferation phase. Endometrial lining will regenerate. So there will be changes in ovarian uterus both, and there will be pituitary hormone and ovarian hormone both will act. Which pituitary hormone? LH and FSH. Which ovarian hormone? Estrogen. Correct. Secretion of gonadotropin. What are gonadotropin? LH and FSH. So they increase gradually. This word gradually is important. They increase gradually, and in biology, these kind of words are more important. Gradually, quickly, correct? Usually, exclusively, these kind of English words are more important during the follicular phase. And there will be follicular development from primary follicle to the mature follicle, and secretion of the estrogen by the follicle. It means ovary have follicle and follicle produce estrogen. Pituitary gland is producing LHF. LH and FSH level will be the maximum in the middle of the cycle. Rapid secretion of only LH will be responsible for LH surge, and this LH surge is responsible for rupture of Graafian follicle and ovulation. So what is responsible for ovulation? LH surge. LH surge. Next, this ovulation will occur on the ovulatory phase 14th day. Here you will have ovulatory phase, 14th day. Ovulatory phase. Next. Followed by the luteal phase, during which remaining part of the Graafian follicle convert into the corpus luteum. That's why luteal phase. Luteal phase and secretory phase. Why secretory phase? Secretion of progesterone. Secretion of progesterone. That's why secretory phase. Yes, Rayan. Hello. Hello. Ji, sir, namaste. Okay. So last phase is progesterone. Progesterone secretory phase, because secretion of progesterone, or luteal phase. Why? Corpus luteum is present. In this case, as you can see, progesterone level is high. Now we want to extend the period. If we want to extend the period, we have to extend the luteal phase. Correct? We have to extend luteal phase, or luteal phase. How we can extend? To keeping by keep giving progesterone, because high level of progesterone, no further bleeding. Correct? High level of progesterone, no further bleeding. Low level of progesterone, then process will start. Okay. So corpus luteum secrete large amount of progesterone. Large word is more important, and this corpus luteum is important for the endometrium maintenance. If progesterone is there, progesterone is responsible for endometrium, no bleeding. No progesterone, bleeding. That's why when female want to skip the period, it means we want to give more progesterone. More progesterone means endometrium will stay. Correct? And this endometrium is really important for the implantation in case of fertilization and other event. During pregnancy, all event will stop, no menstruation. In absence of fertilization, corpus luteum degenerate. Corpus luteum degenerate. Disintegration of the endometrium lining led it to the bleeding, menstruation.
So and for corpus albicans, right?
Yes, Ryan.
So corpus albicans it will be found, no?
Yes. बट एनसीईआरटी में क्योंकि कॉर्पस लुटियम ही दिया हुआ है, तो इतनी ज्यादा हम टेंशन नहीं लेंगे. दिस इज ट्रू. कॉर्पस albicans will form. Period will stop around 50-year age. Menopause. Pause means stop. And if there is a proper regular menstruation, it means normal reproduction. Everything is normal. So female will have periods from the menarche to menopause, because after menopause process will stop. Now look at the diagram. What are the pituitary hormone? LH-FSH. LH-FSH level will be high during follicular phase. Okay. Ovarian event. Developing follicle, mature follicle. What is responsible for maturation? LH, FSH, and ovarian hormone. Ovarian hormone, mainly estrogen. Because as you can see, progesterone level is almost same. Progesterone level is almost same. Correct. Ovarian event, corpus luteum will keep on developing if there is a pregnancy. No pregnancy, corpus luteum will regress. No fertilization, corpus luteum will regress. Done. Any doubt?
सर ये टेबल बता दो एक बार.
कौन सी?
यही वाली.
हाँ, तो इसमें हमें ये देखना है आदित्य, पहले तो सीक्वेंस ऑफ इवेंट. पहले मेंस्ट्रुएशन होगा लगभग 1 to 5 डेज. फिर 5 डेज से लेकर 14th डे तक होगा फॉलिकुलर फेज, सेकेंड फेज, प्रोलिफरेटिव फेज, जहां पे फॉलिकल ग्रो होंगे, है ना. फॉलिकल ग्रो होंगे, और फॉलिकल ग्रो होंगे मतलब प्राइमरी से मैच्योर फॉलिकल. ठीक है. प्रोलिफरेटिव फेज है, क्यों? क्योंकि यहाँ पे जो एंडोमेट्रियम है वो अभी रीजनरेट करना स्टार्ट होगी, प्रोलिफरेशन होगा. फिर ओवुलेटरी फेज होगा थर्ड. ओवुलेटरी फेज होगा थर्ड, जहां पे ओवुलेशन होगा, और ओवुलेशन का रीजन क्या है? एलएच सर्ज. एलएच का लेवल सबसे हाईएस्ट होगा, एलएच सर्ज, है ना. और 14th डे से लेकर 28-29 डे तक क्या होगा? जो ड्यूरेशन है वो है लुटियल फेज, क्योंकि कॉर्पस लुटियम बनेगा. सेक्रेटरी फेज क्योंकि प्रोजेस्टेरोन का सिक्रेशन होगा. ये है फोर्थ में. यहाँ पे हमें अलग अलग चीजें दी हैं, जैसे यूट्राइन इवेंट में हमें ये पता चलेगा कि यूट्रस की एक्टिविटी कैसे हो रही है. जैसे एंडोमेट्रियम लेयर इज मैक्सिमम कम टू द नॉर्मल सिचुएशन ड्यूरिंग द लुटियल फेज. तो लुटियल फेज में क्या यूट्रस के अंदर एंडोमेट्रियम रिकवर होगी फुली, है ना. ओवेरी की बात करें तो फॉलिकुलर फेज में ईस्ट्रोजन का लेवल मैक्सिमम होगा और प्रोजेस्टेरोन का मिनिमम, और लुटियल फेज में प्रोजेस्टेरोन का लेवल मैक्सिमम और प्रोजेस्टेरोन का, ईस्ट्रोजन का मिनिमम. ओवेरी के अंदर क्या होगा? प्राइमरी फॉलिकल मैच्योर बन जाएगा बिफोर ओवुलेशन, और आफ्टर ओवुलेशन पहले तो कॉर्पस लुटियम बनेगा, लेकिन अगर फर्टिलाइजेशन नहीं तो कॉर्पस लुटियम भी रिग्रेस हो जाएगा. ठीक है. और इसी टाइम में एलएच एंड एफएसएच का लेवल पीक होगा ओवुलेशन के टाइम पे, उससे पहले और उसके बाद लेस भी रहेगा. आदित्य.
नहीं सर वो.
So now you can answer this question. (172) कि we want to postpone the bleeding. Postpone the bleeding का मतलब क्या है? We want to extend luteal phase और proliferative luteal phase और secretory phase. Luteal phase और secretory phase, है ना. How to do that? How to postpone luteal or secretory phase? By giving progesterone. Done. Any doubt? Any question? How many statements are incorrect? first read, like before reading the question, na, start reading this. Key restriction endonuclease enzyme, how they will act. है ना? पहला, each of them function by inspecting the length of DNA sequence. ये क्वेश्चन में पूछने से पहले इस बायोटेक्नोलॉजी का चैप्टर अभी हमने किया था ना। बायोटेक्नोलॉजी 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 हाँ So guys, tell me how much I covered. Thank you, Varinder, कॉल करने के लिए. How much like कितना पढ़ाया था मैंने? How much I covered in the last topic?
So you will do the restriction enzyme.
I covered this part. This part fully.
No, sir.
So
Okay.
you start.
I started. Okay. So if I read this again, then as it is given, each restriction enzyme endonuclease inspect the length of the DNA sequence. Okay. So enzyme will read the full sequence. If it find the specific sequence where it acts, it will bind to DNA. Otherwise, it will not bind. It will cut the two strand of the DNA at specific point in the sugar phosphate backbone. Sugar phosphate backbone. Correct? And enzyme will recognize palindromic nucleotide sequence in the DNA. Correct? It recognize only palindromic sequence. समझ में आया सबको? Now solve the question. Where is this? Yes. Priyanka. Each restriction endonuclease function by inspecting the length of DNA sequence. Is this correct? It is. Barinder. One of them recognizes specific sequence of base pair in DNA. Read the same on the two strand when orientation of reading is kept same.
Sir, correct.
Correct. अब इसको हम कंप्लीट ही कर लेते हैं क्योंकि उसके बाद फिर वो अच्छे से हो जाएगा. This part is done. So enzyme will cut the DNA strand at the same site, whether vector DNA or foreign DNA. Same enzyme for both. Correct? It will cut between G and A only. Between G and A only. Between G and A only. When you have get C, GAA TTC, get C, GAA TTC. Very, very, very important. Correct? It will make sticky end in the presence of this enzyme, and both will be joined with the help of DNA ligase enzyme. Sticky end will be joined with the DNA ligase enzyme. What do you mean by palindromic? Form the same word when read in the both directions. But when we say the word palindromic, it read in the both directions. Palindromic in DNA is different. It is not same. Because if we take this GAATTC, if you go in the forward direction, it is GAATTC. If you go backward direction, it will not be GAATTC. So palindromic normally is different from palindromic in DNA. Make sense, guys? You must have realized that. कि palindrome in DNA is different from the palindromic in general. Example, Nitin. If you take Nitin name, Nitin from the forward and backward direction will be same reading. So this is palindromic in English. In DNA, palindromic is different thing. Same thing is here. In DNA, it is the sequence of base pair that read same on two strand when orientation of reading kept same. When orientation of reading kept same. Example, from 5 prime to 3 prime and it is GAATTC. Even in the another strand 5 to 3, it will be same GAATTC. Correct? In opposite side, GAATTC from 5 only. Any doubt? Restriction enzyme cut the DNA strand little away from the center, not far away, little away. Imagine in this topic, little word is more important. Little, and next time maybe away will be more, little away from the center between the same two bases on the opposite strand. You can see here this one GAATTC like this. Little away from the center, as you can see here, not exactly at the center. Little away from the center. Next, this leaves single-stranded portion at the end, correct? And there will be sticky end. अगर sticky end बन जाएंगे, तो they will form hydrogen bond with their complementary counterpart, and they will attach together with the help of enzyme DNA ligase. It will help in the DNA ligase, correct? Correct. When these two DNA will combine together, new DNA will be known as recombinant. New DNA will be known as recombinant. If they cut by the same enzyme, make sticky and join together with the help of DNA ligase enzyme. DNA ligase enzyme. Done. Foreign DNA and vector DNA will use the same enzyme, cut the both strand at specific point, join them with the ligase enzyme, then recombinant DNA form, transform, and the same thing is given here. Source DNA and vector DNA should cut from the same enzyme. Otherwise, you cannot create recombinant vector. You need to use same enzyme. Otherwise, it will not work. If you understood this well, Anvita, tell me this answer. Full topic. First statement correct. Second statement.
True, sir.
बरिंदर ऑलरेडी बरिंदर ने पहले बता दिया था ना शायद. हाँ. Anvita third. They cut the DNA strand far away from the center of the palindromic site.
The fall.
False. Statement was little away from the palindromic sequence, not far away. Then Aditya, these enzymes specifically cut hydrogen bond to get the desired DNA fragment.
True.
So they asked how many are
Three.
wrong. Correct. How many are wrong? Only one is wrong with respect to restriction enzyme.
सर डी रॉन्ग होगा ना?
हाँ, sorry. D with wrong. Correct. Why? D is wrong because enzyme will specifically cut not hydrogen bond but between G and C. It will cut backbone, not hydrogen bond. Backbone. It will cut backbone. It will cut backbone. I'll show you the same statement. This. Look at this. Once it finds specific sequence, it will bind to DNA and cut two strand of the double helix at specific point in the backbone, not hydrogen bond. Cut the backbone. It will cut the backbone at between G and A. Between G and A. So fourth is also wrong. It cut the backbone, not hydrogen bond. It cut the backbone, not hydrogen. Only then sticky end can be produced. Make sense, guys? It will cut backbone. अच्छा किया वरिंद्र बता दिया. So third and fourth are wrong, so answer will be 2. Any doubt, guys?
No, sir.
No, sir.
This colorblind topic is easy, and actually colorblind topic and hemophilia is almost same. So I will do this one more topic, then we'll take break. Okay, 10-minute break, not longer break, because reading biology for long time is very.
Sir, what time of class be there?
Let's see how long it goes.
Because 11 o'clock I have to get ready and by 12 I have to leave.
By 12 you have to leave.
Yes, because I have to be there by 1. They take us into the examination hall 1 hour before the exam.
Where you are going?
Pala.
You have test today?
Yeah.
Today?
Yes, 2 to 5.
2 to 5. Okay, sure. I will show you one more question. See, 164. Who will tell me? Anvita asked. Nirzila. Today
Yeah.
we have more than 900 restriction enzyme isolated. Is this correct? Yes, it is correct. Second, over 20 strain of the eukaryote. First, this is wrong, not eukaryote, bacteria. And we have more than 230 strain, correct? Not only 20, more than 230 strain from bacteria, which recognize same recognition sequence. Of course not. Right? Right? And second one, क्या बोलते हैं, निर्जला. If you have DNA undergo 30 cycle of PCR, it will produce 1 billion copy of this DNA uptake. This is the first part we already did. Second part, PCR will do from the same topic. Quick revision of PCR. First question was from this. We obtain more than 900 restriction enzymes from more than 230 strain of bacteria, not eukaryote. And they recognize different recognition sequence. Right? That's why reading every NCERT word and not making mistake is a big challenge. Then PCR. PCR stand for polymerase chain reaction. Multiple copy of DNA of interest will be synthesized in vitro, means in lab. And in this process, we use 2 set of primer. Question can be asked, how many set of primer are used? How many set of primers are used? Answer is 2. 2 set of primer are used. 2 set of primer are used. Next, what is primer? Primer is small chemically synthesized oligonucleotide, because artificially we are making this chemically synthesized small oligonucleotide, and this is complementary to the DNA. Artificially we are making this sequence, very small sequence oligonucleotide bind to the DNA, and DNA polymerase enzyme we need in this case. Enzyme, what is the role of DNA polymerase enzyme? Enzyme will extend the primer using nucleotide. So primer will use nucleotide and extend the primer. And here genomic DNA, the main DNA, your DNA will be multiplied. Segment of DNA can be amplified, like 1 billion copy will be made in how many cycle? 30 cycle. 30 cycle, 1 billion copy. So this statement is correct. Such amplification is achieved by thermostable DNA polymerase enzyme obtained from bacteria, Thermus aquaticus bacteria. This Bacteria can remain active during very high temperature, and this high temperature will induce denaturation of the. How high? Very high temperature is like 94 to 96 degrees Celsius. This enzyme is active at very high temperature. What is the benefit of high temperature? High temperature will break the DNA, double stranded DNA, correct? And the DNA fragment you can ligate with the help of vector. Sorry, with the help of ligase enzyme with vector. Look at this cycle. First, you have double stranded DNA. You have double stranded DNA. First step will be denaturation. You heat, it will break into the small sequence. Then you add a primer in the process of annealing, correct? And third step, when you add DNA polymerase, enzyme tag polymerase, and deoxynucleotide, extension will happen, and from one DNA, you will get two DNA. It will produce two DNA, and same cycle repeated 30 times. So, how many DNA is produced after n cycle? 2 raised to power n. 2025 NEET question. 2 raised to power n. Done? तो निर्जला, आप मुझे इसी क्वेश्चन का आंसर दोबारा बताओ.
Yes, sir.
So we don't care about the mass of the DNA. DNA will undergo 30 cycle, will make 1 billion copy.
Yes, sir.
है ना? So second statement is correct. First statement is wrong.
सर अगर 50 होता तो कितना होता?
अगर 50 साइकिल होता तो बहुत बड़ा नंबर हो जाता, वो हम नहीं बताते, मतलब वो बहुत ज्यादा इंक्रीज हो जाता, इसलिए
वो पेपर में तो नहीं पूछे जाएंगे सर.
नहीं, वो नहीं पूछा है. कम पूछ सकते हैं कि 2 3 5 साइकिल यू कैन डू, कि अगर इफ 1 साइकिल 2 साइकिल 4 DNA, 3 साइकिल 8 DNA, 4 साइकिल 16, 5 साइकिल, बस इतना ही हो सकता है. 32, 6 साइकिल 64, 7 साइकिल 128, 8 साइकिल 256. अब आगे से कितना ही पूछते हैं. यू कांट डू कैलकुलेशन क्योंकि दिस इज बायोलॉजी नॉट मैथ.
ओके सर.
Rishikesh is there? No. Sarah, tell me this. What is the role of the permease enzyme? We did this from left of the. Any doubt, Sarah? B will be for it. Permease means permission. It will increase the permeability of the cell to the beta galactosidase. So B will be 4. B4, there is only one option. Correct?
Yes.
A will primase, as the name says, it will help in the primer formation in DNA. Primer
Yes.
formation primase. Polynucleotide phosphorylase, enzyme synthesis for RNA. Polynucleotide phosphorylase, ATP addition, energy, and then protein formation. Protein formation. Ribozyme, that we'll discuss in molecular basis of inheritance. Translation, peptide bond formation. Should we stop here for 10-minute break, or you want me to finish translation for ribozyme? Is it okay for everyone, or should I stop here?
Sir, if you
Sir?
continue till when will you take class then?
I will take class 10:30, around 12.
So then can we take a 10-minute break?
Yes. So I'll start at 10:45. Just give me 2 minutes. Aditya, how is it going?
बढ़िया है सर, सही हो रहा है।
Rayan,
Sir, good, sir.
Nizla,
So it was good, but I'm asking that आपने 165 से 170 वाले questions छोड़ दिए?
resume recording. So first question is Rene Descartes, French philosopher, mathematician, biologist. All human knowledge, especially natural science, is directed to development technology. So he said the ultimate purpose of all this science is like how we can apply the values to human being, how we can give comfort to human being. Correct? And this whole thing is anthropocentric. Anthropocentric means human in the main center, and all the technology developed near around the human being. And biotechnology in 20th century is the offshoot of the modern biology. Question can be asked: what is the offshoot of the modern biology? Biotechnology. Correct? Because it increase the qualitative improvement in the health. Quality of the health increase because of that. Food production increase. Correct? Then Herbert Boyer, he obtained in 1969 restriction enzyme from E. coli bacteria. Correct? He used the sticky end of the strand, clipped and sticky end. Correct? And Cohen, he obtains small ringlet of DNA known as plasmid. Ringlet is known as plasmid, and it float on the bacteria. Plasmid float on the bacteria, and together they discovered that these plasmid are responsible for the formation of certain kind of protein, because plasmid is DNA, DNA from RNA, RNA from protein. The whole process is, what is this? Um, Central dogma. Correct? How information passed from DNA to RNA and then protein formation. Central dogma. Second, in biotechnology, we use live organism or enzyme at large scale. At large scale, and we use genetically modified organism. In test tube, we use test tube baby, gene editing, gene synthesis, DNA vaccine, correction of the defective gene, gene therapy. All are the application of DNA. All are biotechnology. Correct? European Federation of Biotechnology give the definition. This definition cover traditional old view and modern view. Correct? And it say biotechnology is where we integrate natural science, nature science, we integrate organism, we integrate cell, we integrate all the thing, so we can get beneficial product for human being. We can get beneficial product for human being. Biotechnology have two main things: genetic engineering, where we change DNA or RNA; bioprocess engineering, where we maintain sterile environment. There should not be contamination. Otherwise, full program is waste. Give me one. Okay, so two part. One, you change DNA or RNA. Second, you maintain sterile environment, so there is no contamination. Correct. And because of this, when we produce antibiotic, vaccine, or enzyme, we make sure there is no undesirable thing. Only desirable microbes or eukaryotic cell should be used in a large quantity. Correct. When we study asexual reproduction, we maintain genetic information. We manage genetic information because copy paste. Sexual reproduction, variation. Old hybridization process, you can bring change by hybridization process, by animal breeding. But the problem with the traditional hybridization is that you get undesirable thing also. Okay, you get this also. What are the component of the genetic engineering? Making a new DNA, recombinant DNA, making multiple copy of DNA, and after making multiple copy, you transfer the gene from one to another. What is the benefit of this? Through this, we can get only one or set of gene, and there is no undesirable thing. Old method, undesirable thing can be present. New method, only desirable. New method, only desirable. Okay. Okay. So, if you see what happened in biotechnology, this most likely DNA cannot multiply anywhere. But if you connect DNA with the recipient gene, means DNA of this cell, you can multiply it in the host organism. Correct? But if you want to multiply your DNA, you have to attach this with specific site known as origin of replication site. In a chromosome, there is a specific DNA sequence known as origin of replication, and this is responsible for initiating replication. Correct? That's why if you want to multiply your DNA in an organism, this should be part of the chromosome which have origin of replication. No origin of replication, no multiplication possible. If we attach alien DNA with the origin of replication, then it can replicate, it can multiply itself in the host organism. Correct? And process of multiplying is known as cloning. Process of multiplication is known as cloning. How we can make a recombinant DNA artificially? So first recombinant DNA that came for the antibiotic resistance gene, where we attach our DNA with the plasmid, native plasmid. This is the definition of plasmid. Plasmid work automatically. Automatically means it is not dependent on the main DNA. It can replicate. Plasmid is a DNA which is circular in nature. Plasmid is the extra DNA of the bacteria. Which bacteria? Salmonella bacteria. If you remember, I asked this question from someone, I don't remember, and same question was there in test. So, Stanley Cohen and Boyer in 1972, Singer and Nicholson experiment 1972, they isolated antibiotic resistance gene by cutting the DNA from the plasmid, and this is responsible for the antibiotic resistance. Correct? Responsible for antibiotic resistance. So they discovered, Cohen and Boyer discovered the plasmid and the whole process by using Salmonella typhimurium. Now look at this question. Nirjala,
Yes, sir.
165.
Option 1.
Yes. Construction of the first recombinant DNA emerged from the possibility of linking gene encoding antibiotic with the native plasmid of that question. Same topic. Right? And same thing was given here. See, first recombinant DNA emerged from the possibility of linking the gene encoding antibiotic with the native plasmid of Salmonella typhi. Exactly same line. We can cut the DNA at specific location with the help of molecular seizure known as restriction enzyme. This can be linked like with the plasmid DNA. Plasmid can act as a vector. It transfers the DNA. Right? Like how mosquito can act as vector. Correct? Mosquito is a vector for the malaria parasite. Same way plasmid act as vector because plasmid deliver outer DNA into the host link. And if you link the antibiotic resistance gene with the plasmid vector, it is possible with the help of DNA ligase enzyme. So what is the role of ligase enzyme? Attach plasmid DNA and alien DNA together. Correct? And this will act on the cut part of DNA and join their end. And this make a new combination of circular plasmid, which is produced in vitro. In vitro means lab. In vitro means it was produced in lab, and this is known as recombinant DNA. Correct? When this DNA is transferred into E. coli, then bacteria, E. coli is like nature of E. coli and Salmonella is exactly same. They belong to same family. Both are gram-negative bacteria, so they're very close to each other. So it can replicate using no a new host DNA polymerase enzyme because bacteria have DNA polymerase enzyme. Bacteria can multiply the plasmid. Bacteria can multiply RDNA and make multiple copies. Right. So when you make multiple copy, that process is cloning. That process is cloning. There are 3 steps in this process. First, you identify the right DNA. Question can be asked, write the sequence of this. So identify the right DNA, introduce this DNA into the host, and last, maintenance of introduced uh, DNA into the host. See, first, you will take one proper DNA. You will introduce this DNA in bacteria, and you will maintain this DNA so same thing is transferred into the new cell. New cell should have this same thing. Okay? So introduction part done, this part done, this whole thing done, small gel electrophoresis is left. Huh. Now, because recently I taught this, all of you take 5 minutes, read this, and then I will ask you question. Okay? Because it will be boring if I teach same thing again. You read for 5 minutes, ask your question, then I will ask you a question from the same topic.
From whole chapter or gel
No, this topic, gel electrophoresis. Done, guys?
Sir, 2 minutes.
Done? Do I ask question? And answer quickly, because NCERT will be visible to you. So you can revise it again instantly. First, Anvita. Agar gel is obtained from
So, seaweeds.
seaweed, natural polymer. Varinder. DNA fragments separate according to size. Through which effect?
तो वर्ड
Sheaving
एक
effect.
बार रिपीट कर दीजिए.
शेविंग इफेक्ट प्रोवाइडेड बाय अगारोज जेल की हम साइज के बेस पे सेपरेट करेंगे डीएनए बैंड को, तो इफेक्ट कौन सा होगा? शेविंग इफेक्ट. निर्जला.
Yes, sir.
लार्जर द फ्रेगमेंट साइज.
Larger the fragment size, तो it
मिनिमम
means कि वो
होगा.
minimum, हाँ, it means कि वो उसके पास ही आएगा.
हाँ. सारा. व्हाट इज द चार्ज ऑन डीएनए एंड टुवर्ड विच इलेक्ट्रोड इट विल मूव?
It's a charge on DNA is negative.
So it will move toward
Anode, sir.
anode. So where we keep the DNA? Cathode side or anode side?
So cathode side.
Cathode side. Manan, what is the sequence of step of staining and UV? पहले किसको करेंगे, बाद में किसको करेंगे?
ट्रेनिंग और
यूवी एक्सपोजर में से फर्स्ट स्टेनिंग.
पहले ट्रेनिंग करेंगे,
पहले स्टेनिंग, फिर
फिर
यूवी,
UV।
फिर ब्राइट ब्राइट ऑरेंज कलर,
हाँ,
फिर
ब्राइट ऑरेंज कलर आएगा जो
कट आउट
इथिडियम
डीएनए.
ब्रोमाइड के स्टेन्ड उसके अंदर आएगा, वो जेल के अंदर।
कट
फिर वो
आउट डीएनए.
उनको अलग अलग करेंगे, फिर एग्रोस जेल के अंदर जो अलग अलग करते हैं काट के उन्हें, और फिर उनको चार्ज की मदद से, मतलब उनके मॉलिक्यूलर बेस की मदद से उनको उसमें प्लेस करेंगे वेल्स के थ्रू।
करेक्ट. नेक्स्ट. It is which lane will be digested and undigested?
सॉरी सर।
Which lane will be digested and undigested? Digested 2 to 4, undigested 1, because biggest. No movement, undigested. Movement means digested. No movement of DNA, undigested. Movement of DNA. Hello. Any doubt, guys? Anybody have
पॉइंट
question?
कहाँ लिखा है?
क्या अनिता?
ये वाला पॉइंट कहाँ लिखा है?
ये लिखा हुआ. Typical agarose gel electrophoresis showing migration of undigested lane 1. Undigested means when there is no movement. And digested from set 2 to lane 2 to 4. It means there is movement. 2 to 4 there is movement. In 1 no movement. That's why undigested. देख रहे हो, एनसीईआरटी को पढ़ने के बाद भी कई बार हमें ये नहीं पता होता कि अच्छा ये कुछ इंपॉर्टेंट, ये ट्विस्ट कर सकते हैं क्वेश्चन को.
सर एक चीज पूछना है, ये वेल्स के अंदर डालेंगे या वेल्स के आगे डालेंगे इन्हें?
वेल्स में डालेंगे. वेल्स कुछ नहीं होता, बस खाली जगह होती है, फिर उसके उसके अंदर मूव करने लग जाता
ओके सर।
है. नेक्स्ट. Read this topic, guys. Read this.
Sir, competent host वाला.
तो ये भी रीड करके आप फिर क्वेश्चंस पूछेंगे?
हाँ guys, are you getting my voice properly? Is there any issue?
सर कोई
No,
इशू
sir.
नहीं आ रही आराम से।
Okay, done.
एक मिनट बाद।
हाँ
Sir, I'm done.
Okay, so let me check who were left in the last class. Aditya, Rehan, and Chandana. Aditya, what is the
Yes,
charge on, what is the nature of DNA? Hydrophobic, hydrophilic? Why can't it pass through the cell membrane?
सर हाइड्रोफिलिक होता है वो.
हाइड्रोफिलिक होता है तो क्यों नहीं पास कर सकता सेल मेम्ब्रेन से? बिकॉज सेल मेम्ब्रेन मेड अप ऑफ लिपिड बाइलेयर, तो वो काइंड ऑफ मेड अप ऑफ फैट, so it cannot allow, like plasma membrane or cell membrane cannot allow to pass through it because it is mainly made up of lipid bilayer, है ना? So next, Chandana, what is the role of calcium in competent cell?
So it increases the efficiency for DNA to enter into the host.
How? Because it make more pore in the cell wall.
Yes.
Rayan, what is heat shock method?
Sir, can you again repeat?
Heat shock method. First, recombinant DNA incubate, recombinant DNA is incubated with the cell. So first on ice, then 42 degrees Celsius temperature, then ice. So bacteria will take recombinant DNA. Heat shock method. Anvita, what is biolistic or gene gun method?
सर ये प्लांट के लिए यूज होता है. इसमें लाइक हाई वेलोसिटी वाले पार्टिकल्स देते हैं, जैसे कि गोल्ड या टंगस्टन, उसको, और इसमें डीएनए भी लगा होता है, लाइक कवर्ड होता है, फिर उसको बम्बार्ड करते हैं तो वो डायरेक्टली लाइक एंटर कर जाता है
हाँ.
प्लांट के सेल्स में.
हाँ. तो नेक्स्ट,
अब क्या करना है?
इसको रीड करना है.
अच्छा, ओके. तो मतलब बेसिकली बचा चैप्टर ना, क्योंकि इसके सब पार्ट्स हैं.
हाँ.
तो अब डाउनस्ट्रीम प्रोसेसिंग तक.
नहीं, मैं वन बाय वन लूंगा, जो स्क्रीन पे विजिबल होगा वही तक.
अच्छा, ठीक है.
What happened, Jordan? Jordan, what happened?
Yes, he's texting on the chat thing.
Sir, I thought I would attend class. Great. I am on a bus. I can't speak right now. No issue. You have, I am showing one topic. You have to read that topic and I will ask question. I will ask question from other. You can listen and it will be beneficial. Good job, Jordan. Dedication. Done, guys. So, basic is, tell me all these steps happen in recombinant DNA technology in sequence. Basic is, are you there?
No, sir, he's not.
Then Nirdala.
Yes, sir.
Sequence of this step in recombinant DNA technology.
सबसे पहले isolation of DNA.
Correct. Second.
फिर दूसरा है fragmentation of DNA by restriction endonucleases.
Yeah,
तीसरा
third.
है isolation of a desired DNA fragment like और चौथा है ligation of the DNA fragment into a vector. फिफ्थ है recombinant DNA into the host. और सिक्स्थ है culturing the host cells in a medium at large scale. और सेवेंथ है extraction of the desired product.
Next, Ryan. What are the different enzymes we use to destroy the cell wall of the organism?
So one is lysozyme for the bacteria, then the chitinase for the fungus, and the cellulase for the plant cells.
Yes, Aditya.
Yes, sir.
To obtain the pure DNA, what do we do? Like how to remove other molecule, RNA, protein, how to settle DNA?
क्वेश्चन रिपीट करना सर।
तो सबसे पहले तो कि हम अलग कैसे करें आरएनए और प्रोटीन को, और दूसरा कि डीएनए को सेटल कैसे करें।
सर अलग करने के तो राइबोन्यूक्लियस चाहिए।
RNAs to remove RNA and protease to remove
प्रोटीन
protein.
प्रोटीएज फिर चिल्ड इथेनॉल में डाल।
फिर इथेनॉल कर देंगे तो प्योर डीएनए अल्टीमेटली प्रेसिपिटेट हो जाएगा, और स्पूलिंग प्रोसेस से हम उसको रिमूव कर सकते हैं, स्पूलिंग प्रोसेस। एंड वो डीएनए एक फाइन थ्रेड की तरह दिखेंगे इन सस्पेंशन, है ना। दिस इज ऑलरेडी डन. The restriction enzyme digestion are performed by incubating the pure DNA molecule with a restriction enzyme. Optimum condition, agarogel electrophoresis to check the progress of enzyme. Correct. Agarogel electrophoresis to check the progress of the restriction enzyme digestion की अच्छे से हुआ है कि नहीं. DNA negative charge move toward the positive anode. Correct. And same thing is repeated for the vector DNA also. Correct. Then we have to join them. We have to get the gene of interest from the source DNA and then add ligase enzyme, correct? And there will be proper recombinant DNA. PCR done. There are multiple methods how we can add DNA into the proper cell, and we did the competent cell for bacteria heat shock method, for plant bioelastic or gene gun method, for animal micro injection or disarm pathogen, है ना? Correct. So if recombinant DNA is having antibiotic resistance gene is transferred into E. coli, host cell will convert into the ampicillin resistance gene. मान लो हमारे पास है एक recombinant DNA and this recombinant DNA have ampicillin gene. If we transfer this recombinant DNA with ampicillin gene in E. coli bacteria will become resistant to the antibiotic, क्योंकि अब हमने एंटीबायोटिक रेजिस्टेंस जीन इंट्रोड्यूस कर दिया है ना। तो ट्रांसफॉर्मेंट तो जो होंगे जिसके अंदर वो चला गया होगा वो तो एम्पिसिलिन में सर्वाइव कर जाएंगे, नॉन ट्रांसफॉर्मेंट खत्म हो जाए, है ना। तो विल गेट द आइडिया कि विच इज अ सिलेक्टेबल मार्कर। तो एम्पिसिलिन रेजिस्टेंस जीन इज सिलेक्टेबल मार्कर इन दिस केस, right? Gene is selectable marker. Gene is selectable marker. Yeah. Last, very basic process. Next, you have to read. I'll explain this. When we add DNA into the cloning vector in different cell, because we want to multiply the DNA. In this case, ultimate goal is to get best protein, correct? And for best protein, we want कि recombinant DNA should express itself properly, correct? So when recombinant gene will express itself in the proper situation, then we need to maintain the proper condition, optimum condition, best condition, so it can multiply, right? On the large scale, on the large scale. So if any protein encoding gene is expressed in the host, it is heterologous host, and protein will be known as recombinant protein. Why heterologous? Because you have host with the different DNA. That's why hetero means different nature. Correct? Cell we can grow in the small scale. We can use large scale system. Large scale system is continuous culture system, where we use medium drain out from one side and fresh medium is added on the another side, like continuous process. Continuous process. Correct? Today we read from the plant growth and development that there is active low or exponential phase where bacteria will continuously divide, correct? And it will produce large biomass and high yield of our product, correct? For like small volume is not sufficient, so we need large quantity. For large quantity, we need bioreactor. What is bioreactor? Bioreactor are the containers where you can grow at the large scale. You can grow the bacteria at large scale, correct? Where we can add raw material and convert into specific product, correct? We can use microbial plant, animal, human cell, anything is possible, correct? What is the benefit of bioreactor? In bioreactor, we provide the best condition. And most common bioreactor is stirring type, like continuously we provide all the system. Look at the diagram, we recently did this. Simple, where lowermost start from here. First motor, second foam breaker, third blade, flat blade impeller, fourth cultural growth, fifth sterile air, sixth steam for sterilization, and seventh acid base pH control. Uppermost pH control, side steam for sterilization, lowermost air, correct? This side we increase surface area. Why? More surface area, more oxygen transfer. More area, more oxygen transfer. And if you see bubble will increase oxygen surface area. More surface area, more growth of the bacteria. Gas entrainment means more gas is present, more oxygen means more bacteria growth. Right? Second one is known as sparse type, proper mixing, more oxygen. And this is simple one. Correct? Sparse means more advanced because sterile air bubble are sparse in this case. Correct? So next in reactor, what is the shape of the sterile tank reactor? Cylindrical shape. Base, curve base. Benefit, proper mixing of the content. What happen when there is sterile type, there is a proper mixing, proper oxygen availability. Correct? We can also add more air bubble. Example, if you remember in cold drink, when we put cold drink, there is a lot of air bubbles. Same thing. Correct? So bioreactor have agitator system for mixing, oxygen delivery, foam control, temperature control, uh, pH control, sampling port, because you want to check sample ki everything is acting perfectly. Is there any issue? Downstreaming process. Once everything is done, we have to get the product. We have to pack the product properly so we can do the marketing. We have to give finishing product. So that is the downstreaming process. Separation of the final product, purification of the final product, and packaging of the final product. Correct? Separation and purification. So sometimes we can add proper preservative in the product if need. If it is medicine, drug, medicine, then we have to do clinical trial also. Correct? Proper quality testing is important for each product. And downstreaming process plus quality control testing can vary from product to product. It is not same. Quality index is different. In medicine, they're like more strict trial, but in case of you know, for some simple process for plant, process will be little bit easier, and every single thing is done. Now you can ask कि सर आपने ये चैप्टर स्पेशली क्यों करवाया? Because NEET 2025 NEET 2025 में there were 6 questions from this chapter. Correct? 6 questions. 6 means 24 marks. Imagine we did 4 chapter of physics, like modern physics 4 chapter. Dual nature, atom, nuclei, 4 chapter. Total question from these 4 chapter 6. Only from biotechnology 6. So what is a smart move? To do biotechnology multiple times. From biotechnology application, 1 question. Make sense, guys? Why especially we focused more on this chapter? Any doubt?
Yes,
No, sir.
So it is 12:15. Should we take break?